Hi friends, myself Dipankar Majumdar. Welcome to my channel Microbiologist's Forum. Today I will discuss about the most required and basic instrument to study the microorganisms, that is the microscopy. Sight is human's dominant sense and the sensory world is primarily a visual one. Magnifying lenses, microscopes, telescopes provide extension of human's visual sense. Viewing or seeing depends mainly on the resolving power of the human eye. Resolving power is nothing but the power to separate two very closely associated points as two separate points. Okay, and the word microscope is derived from two Greek words. Micro, M-I-K-R-O, micro means small, and scopia, S-C-O-P-I-A. Scopia means to see. That is the instrument which is used to see very tiny or small objects, and the science that deals with the manufacture and functioning of the microscope is known as the microscopy. Microscopy are mainly of two types, light microscopy and electron microscopy, on the basis of the source of illumination. That is, when we are using light, that is mainly the visible light, is simply known as the light microscope. When we are using electron to resolve the tiny objects in microscope, is known as the electron microscope. And today I will just briefly discuss about the structure and function of the compound light microscope. Compound light microscope consists of more than two lenses, so it is so called compound microscope. Okay, and the light which is used in the microscope is visible light. We should look at the properties and types of the lights which is used. Okay, this is the basic structure of the light, that is the wavelength, what is crest, what is amplitude, etc. And the light, the visible light which is used in the microscope is electromagnetic in nature. It is simply known as the electromagnetic radiation. That means it is having an electric field as well as a magnetic field, and the direction of propagation is shown in the slide. This is also the electromagnetic wave, and it is the light that is mainly the visible light or white light which is dispersed into 7 light spectrum when it is passing through a glass prism. It is simply known as the visible light spectrum. From lower to higher wavelength, it is violet, indigo, blue, green, yellow, red and orange. This is the electromagnetic spectrum from right side to left side, from radio wave to gamma ray. The wavelength of light is decreasing, but the power of resolution is increasing. If you see the diagram, you can easily understand that to resolve atomic nuclei, gamma rays which are very shorter in wavelength are used, and to see human beings or buildings, in that case radio wave or microwave can be used. Okay. So, when the wavelength of light is decreasing, the limit of resolution is increasing. Another interesting thing is that when the wavelength of light is decreasing, the tissue damaging power of the radiation is also increasing. This is the structure of electromagnetic spectrum, and this is the basic component of the light microscope. If we see the components of the microscope, this is mirror, then stage, mechanical stage, then objective lens, then body tube, then eyepiece lens. Along with these, fine adjustment screw, coarse adjustment screw, etc. are there. So, we can divide the parts of the microscope into two parts. One is the optical part, another one is the mechanical part. In case of optical part, mainly these are made up of glass. In case of optical parts, the first part is the mirror. Mirror of the microscope is basically plano-concave in nature. Plano-concave means it is a lens that is having one side plane and one side concave. You can ask why it is plano-concave. It may be biconcave or biconvex or both side plane, etc. But this is used due to some speciality. When we are using the sunlight as the source of illumination, then the concave side is used because we need to gather the light ray and then to form the beam of light. For this purpose, concave side is used. The beam of light is formed, then it is passing through the condenser, and then condensation of the light rays are occurred, and then it is passing through the specimen and forming the image, and after that it is passing through the objective lens and does the magnification, and then also passing through the ocular lens and then further magnify the image. After that it enters into the human eye. For that purpose, concave side is used. When we are using the electric bulb as the source of illumination, it is an artificial form of light, then we should use the plain side because the source of light is very much nearer to the microscope. At that time, plain side is used to reflect the light through the condenser and then form the image. Okay. And this is the basic diagram in the right side of the slide, the basic structure of the microscope, which is mostly used in the laboratories. Okay. Now, these are the types of lenses. Lenses are mainly of two types, positive lenses and negative lenses. Positive lenses are mainly three types: double convex or biconvex, plano-convex and convex meniscus. As well as the negative lenses are also three types: double concave, plano-concave and concave meniscus. In case of microscopic mirror, the lens is plano-concave. Now, coming to the resolution. What is resolution? I have just previously discussed that it is the power of a lens to separate two very closely situated points as two separate points. In the bottom of the diagram, two circles are there. They are separated by some distance. it can be resolved by our naked human eye. Our naked human eye can resolve 0.2 millimeter, that is 200 micron our human eye can resolve. That means when 2 points are separated by a distance of 200 nanometer, then our naked eye can resolve these 2 points as 2 separate points. If it is 50 micron, then there is still a distance, but our human eye is not able to distinguish the points as 2 separate points. At that time, we will see the points as 2 overlapped points. It is beyond our resolution, power of resolution of the human eye. For that purpose, we should need some aids. Microscope is one of such aid that helps us to increase the power of resolution. The lower the wavelength of light is used, the better the power of resolution, and this is defined by Abbe's equation. This is the Abbe's equation. D is the power of resolution, lambda is the wavelength of light used, by 2 Na. Na is the numerical aperture, and Na is equal to n sin theta. N is the refractive index of the medium, and theta is the maximum half angle created by the objective lens with the specimen plane. The maximum value of theta is 90 degree, but it will not be created practically. We will come to that point in later. This is the Abbe's diffraction limit from right side to left side, or you can see from left side to right side, and here mammalian cell, bacteria, mitochondria. These can be resolved by light microscope, but virus, protein, small molecules can't be resolved by light microscope. It can be resolved with the help of electron microscope because it is beyond the Abbe's resolution limit, that is 0.2 micron. Now coming to that point, the numerical aperture of the microscope. Okay, here theta is the half of the angle, that is half of the cone that is created with the objective lens and the slide with specimen, and the value of the theta is half angle. In the left side diagram, the value of theta is lesser, and in right side the value of theta is greater, because the lens in the left diagram, the lens is quite large distance from the specimen, so the angle value of theta is lesser. In that case, in right side direction, right side diagram, the theta value is higher because the lens is much nearer to the specimen, and this is due to, if you look at the objectives of the microscope, there are various types of microscopes are present: 4x, 10x, 40x and 100x, and 4x is very much smaller in diameter, smaller in length, then 10x is larger than 4x, then 40x x means magnification, power of magnification, and then 100x. 100x is larger in length. Why 4x is smaller and 100x is larger? This is due to the number of elements of the lens. A lens with higher magnification can be created or can be manufactured by the combination of the small lenses. The lenses may be plano-concave, may be biconcave, biconvex, etc. By combining that small lenses, the lens of a larger magnification can be created. For that reason, the 100x objective is having large number of elements of the lenses. 4x is lesser number of the element of the lenses. For that reason, 4x is smaller in length, 100x is higher in length, and 100x is very much nearer to the specimen in length. For that reason, you can use immersion oil with the 100x lens. You will not be able to use the immersion oil with 4x or 10x or 40x, because the distance between the specimen and the objective lens in 3 lenses is higher. So, you will not be able to fill the gaps with the help of the immersion oil. And immersion oil is an oil, basically it is cedarwood oil, that is having the same refractive index of the glass, that is 1.51. So, in case of 100x lens, the gap which is very much smaller between the lens and the specimen can be filled with immersion oil. So, the gap with immersion oil is having the same refractive index with the glass. Specimen slide is made up of glass, lens is made up of glass. So, there will be lesser chance of refraction of light due to the changing of the medium. But in case of 40x, it is not possible to use oil immersion because the gap is higher. So, it is filled with air. The refractive index of the air is 1. The refractive index of water is 1.33. The refractive index of glycerin is 1.46. But the cedarwood oil is having the refractive index of 1.51, that is equal to glass. If the refractive index of the medium is not changed, the refraction of the light will not be occurred. So, in case of 100x oil immersion objective, the image is much more brighter along with the use of immersion oil. It fills the gap, cuts the chances of refraction, as well as it lowers the chances of mechanical friction. For that reason, along with 100x objective, immersion oil is used. But in case of 4x, 10x or 40x, the immersion oil is not used. In that diagram, you will be able to see very clearly that immersion oil is used in between the cover slip of the specimen and the objective, and there is no chance of refraction in between the specimen slide and the objective because it is filled with immersion oil. So, the chance of loss of light is lesser. For that reason, the image will be much more brighter, the quality of the image will be superior. And in case of oil immersion objective, the maximum numerical aperture is 1.4, but maximum theta value theoretically is 90 degree, and according to the Abbe's equation, numerical aperture Na is equal to n sin theta. N is the refractive index of the medium. In case of oil immersion objective, it is 1.51, and the theta value, maximum theta value is 90 degree. So, in case of 90 degree, the value of sin theta, sin 90 is 1. So, the numerical aperture for an oil immersion objective should be 1.51, but it in practical doesn't exceed 1.4, because in practical you will not be able to create the cone of the angle, that is 180 degree. If it is not possible, then theta will not be 90 degree, it will be always lesser than 90 degree. For that reason, the numerical aperture for oil immersion objective is 1.4, and in case of dry objectives, that is 4x, 10x or 40x, maximum numerical aperture is 0.96, because the theta value is lesser and AR is used, or sorry, AR is present. You will not be able to cover the gap with any other material except AR. So, the maximum numerical aperture value in case of dry objectives are 0.96. And another thing is, you will not be able to make more than 100x objective. In the next part, I will discuss about this. Thanks for patience hearing. Thank you.